So, the social brain in health and disease. Humans, like many mammals, are an intensely social species. We experience social interactions as rewarding from infancy, and the social skills we develop in the context of our earliest interpersonal relationships are critical for survival, group cooperation, and personal well being. Social isolation or loss dramatically increase the risk for depression, anxiety, substance abuse, and personality disorders like borderline personality disorder. In several disorders, like autism spectrum disorder, impaired social functioning is the defining core feature. So today we'll be speaking about autism and some of the biological substrates that will connect my talk to other talks. So autism is a significant public health problem. It's been rising in prevalence and now impacts 1 in 36 US children. Over an individual's lifetime, on average, it costs $3.6 million in terms of social costs. Autism is diagnosed on the basis of 2 core behavioral features. The first is persistent difficulties in social communication and interaction. So failure to initiate or respond to social interactions, deficits in eye contact, inability to understand other people's emotions, or absence of interest in peer relationships. The second diagnostic feature of autism is the presence of restricted, repetitive patterns of behavior, interests, or activities. Things such as lining up toys, adherence to rigid routines, and having highly restricted interests, such as trains. Autism impacts 4 males for every 1 female, and social symptoms are widely viewed as the most debilitating symptom of the autism spectrum. Unfortunately, the biological basis of autism remains poorly understood, and that's going to be one of the topics that we'll be discussing today. So when I began doing research in this field, I was curious about whether the neurobiology that regulates social behavior could provide insights into the biological basis of autism. And so, as you've heard from other speakers in this symposium, there are 2 key molecules that are involved in regulating social behavior in virtually every mammal studied. And these are oxytocin and vasopressin. They're 2 very small peptides that are made in a part of the brain called the hypothalamus. And when they're secreted into the blood, oxytocin's involved in orchestrating childbirth, so uterine contractions, and then also during breastfeeding, the process of letting milk down into the breast. Vasopressin, which is structurally related to oxytocin, they only differ by 2 amino acids, has been implicated in fluid balance and blood pressure. But oxytocin and vasopressin also have neural projections throughout the brain. And so this posed, decades ago, we wanted to know, what is their purpose in the brain? And so there was a zoologist at Duke University in 1971 who wrote an article called Mother Love, What Turns It On? And this is really what launched the field of the neurobiological basis of social behavior. And so what we now know is that oxytocin orchestrates, during the process of childbirth, maternal care in goats, sheep, and many different rodents studied. And particularly in rats, there was some interesting work done in the late 70s or early 80s showing that about 18% of rats are maternal before they've had babies. And that the process of childbirth sort of induces maternal care. But what researchers found was that you could actually give an oxytocin injection directly into the brain of a rat and actually turn on maternal behavior. So thus launching this field of the neurobiological regulation of social behavior. And then moving past maternal care, work done by Sue Carter, one of the speakers in this symposium, showed that prairie voles, which are a very social rodent species, that oxytocin infusion induces these monogamous pair bonds, and as compared to this non-monogamous species, where you can give oxytocin and it doesn't induce a pair bond. And what was interesting was very early work in this field showed that vasopressin, this sort of sister hormone of oxytocin, was what was critical for this pair bonding behavior in prairie voles, as well as paternal care of young. And so when I entered this field, I was very interested in asking the question of does the neurobiology that regulates social behavior, can it provide insights into autism? And so is it possible that impairments in these signaling pathways could provide a sense of where we could be looking for medications to treat social functioning difficulties in people with autism? And so I'm going to just briefly go over the research roadmap. So the work I'll be talking about today is work we've done to develop an animal model to better understand the biological basis of social impairments. And this is in rhesus monkeys. And then to test if the biological findings that we found in monkeys translate to people with autism. And then I'll tell you a little bit about work that we did testing a drug to improve social functioning in people with autism. So rhesus macaques are a species of monkey native to Asia. They demonstrate complex social cognition abilities. They use vision as a primary sensory modality. And we know that they have large individual differences in social behavior. And we know from past work that they show naturally occurring social impairments. And so what I was interested in doing was asking at the California National Primate Research Center, which is about 100 miles from Stanford, was could we look at this large population of rhesus monkeys and could we develop an algorithm to identify the social extremes of this population to be able to study this naturally occurring social behavior deficits and then look at the underlying biological substrates of it. And I'm going to sort of fast forward over about 10 years of research to talk on my next slide about these naturally occurring low social monkeys that have features directly relevant to human autism. So what we know in studying these animals is that using rating scales derived from a human autism trait measure, that low social male rhesus monkeys have greater number of autistic-like traits, that they show abnormalities in perception and reaction to social stimuli, including face recognition deficits, whereas object recognition is intact. We've shown that these animals in their large outdoor home corrals show impairment in reciprocal social interactions. So they show decreased affiliation and grooming and decreased initiation, but not receipt of pro-social behavioral overtures. We've done medical record reviews of these monkeys and shown that they have greater traumatic injuries and bullying by their peers, which worsens with autistic trait number, which is a phenomenon that's also been documented in people with autism. We've shown that these behavioral difficulties have an early onset. So we've been able to identify subtle social information processing deficits in infancy that predict low sociality later in adulthood. And we've also been able to show that these behaviors are heritable, as well as the number of autistic-like traits are also heritable, which is something that we also see in people with autism. So then the next question we asked was, how do we biologically detect these social deficits? So what we know is that autism is a brain disorder, and we could look for the biological basis in a number of different fluids. One could be blood, and another one could be cerebral spinal fluid, which is the fluid that braids the brain and spinal column. Most work in humans has been done in blood, where scientists have looked for biological markers that indicate a disease state. That's what a biomarker is. But unfortunately, the vast majority of work that's been done in biomarker discovery efforts in blood has shown poor detection and poor reproducibility. And we got interested in looking at cerebral spinal fluid because it's more representative of brain biochemistry than is blood. And cerebral spinal fluid has been extremely useful for detecting other brain diseases like various forms of dementia, as well as multiple sclerosis. And so the question we next posed was if we were able to look for biomarkers in CSF and blood, could we differentiate low social from high social monkeys? And so that's what we did. We basically looked for key molecules in cerebral spinal fluid and blood samples, including oxytocin and vasopressin. Recall, these are these social neuropeptides that have been implicated in maternal behavior, monogamy, paternal behavior. And what we wanted to ask was, were these molecules low in these rhesus monkeys? And what we found, if you look at the graph here on the left, the dashed line is a 50% probability of being low social. And so the blue dots that are above the dashed line are individuals that are correctly classified. And orange dots below the dashed line are individuals that are correctly classified. And the way to interpret this figure is that individuals that are low social above the dashed line are correctly classified. So basically, knowing the CSF vasopressin concentration alone allowed us to say that a monkey was low social. And in the next graph over, you can see that there were highly significant group differences, such that the low social monkeys have lower levels of vasopressin and cerebral spinal fluid. We also then showed that vasopressin levels were tightly linked to the time spent in social grooming, which is a critical behavior that many primate species use to cement and maintain social bonds. And then we took multiple CSF samples from individual monkeys. And what we showed here is that on the x-axis are individual monkeys, on the y-axis is the vasopressin level in spinal fluid, and that there's a lot of variability between monkeys, but within each monkey, there is a trait-like vasopressin level, suggesting that vasopressin is a biomarker within an individual, and it's a robust biomarker of not only group differences, but also social behaviors that are, critical for these social bonds in this species. We were able to also show that if we'd only looked in blood, we would not have found these group differences between low social and high social monkeys. And interestingly, we found no group differences in either blood or cerebral spinal fluid levels of oxytocin, the sister hormone of vasopressin. And what was pretty interesting about that to me was that I mentioned previously is that in autism, it's a extremely male biased disorder. And what was fascinating was that in the early days of this work, vasopressin was the molecule that had been implicated in male prosocial behavior. And here we're showing that it's reduced in rhesus monkeys that have social impairments with direct relevance to autism in people. So the next question we wanted to ask was that we had identified CSF vasopressin level as this key indicator of impaired social functioning in low social monkeys. And what we wanted to ask was, does this biomarker finding translate to people on the autism spectrum? And so we launched a proof of concept study where we were able to get spinal fluid from people with and without autism. And what we were able to find, again, much like in this figure labeled A, is that by just knowing the CSF vasopressin level, we could correctly classify 13 out of 14 individuals, much like we could with the low social monkeys. We once again showed that vasopressin levels were markedly reduced in individuals with autism. And then we followed this up with a replication cohort. So this is one of the largest cohorts studied to date, where we were able to look at cerebral spinal fluid levels of both oxytocin and vasopressin in children with autism. And in this instance, we were able to show to date, we had only looked at male rhesus monkeys and male children with autism, but here we were also able to include female children with autism. And in both males and females with autism, we were able to replicate this finding that vasopressin levels in cerebral spinal fluid were once again markedly reduced. And in this case, because these children had been very well characterized, we were able to show that the lower the cerebral spinal fluid vasopressin level, the greater the clinical symptom severity in this population. Once again, we saw no group differences in cerebral spinal fluid levels of oxytocin, which was actually pretty interesting. A, because we were able to replicate it, but B, even including females where oxytocin had been more tightly linked in these early research, vasopressin was what was reduced in both males and females with autism. So our next step was to ask, is CSF vasopressin a marker of risk for autism? And so autism can be reliably diagnosed by about 2 years of age. And so what we wanted to ask was, is this autism biomarker evident very early in life before symptoms first manifest? And so what we were able to do was there were a couple thousand banked spinal fluid samples from human infants in the first couple months of life, where spinal fluid was collected during standard of care medical procedures. And then what we did is we looked at the spinal fluid from children that ended up being healthy. And so this is a reasonably healthy sample. And then we just followed them across development in the medical records till they were 12 years of age. And then we were able to identify who went on to receive an autism diagnosis and who developed typically. And what was very interesting was in the first month or two of life, well before behavioral symptoms for autism first manifest, we saw that these children that later received an autism diagnosis had low levels of vasopressin and cerebral spinal fluid compared to control children. And interestingly, we did not see any differences in oxytocin levels in cerebral spinal fluid, suggesting again that these effects were specific to vasopressin. And then we also looked in a relationship between blood and CSF levels. So we know later in life, blood vasopressin levels are not well correlated with CSF vasopressin levels. But in this early neonatal period, in this separate sample, we're able to show a very nice correlation, suggesting that if we wanted to develop clinical surveillance tools to be able to identify individuals at risk or individuals that could be prioritized for clinical appointments, we might be able to do this in blood. And so this is an area that's under active investigation, and we're hoping to be able to replicate this across multiple sites and follow children from birth until, the first into early childhood. So this work also suggested a brain pathway of interest for developing novel medications to treat social difficulties in people with autism. And so specifically, this led us to hypothesize that brain signaling may be impaired in the brains of children with autism. And so we posed the question, does vasopressin treatment enhance social abilities in people on the autism spectrum? And we know from work done over 20 years ago now that if we administer vasopressin intranasally, that it can penetrate cerebral spinal fluid, suggesting that it achieves access to the brain. And we know from some single-dose intranasal vasopressin studies in adult male healthy humans that vasopressin can enhance memory for social information, identification of social words, and can induce cooperative behavior. But no prior research had tested the safety and effectiveness of intranasal vasopressin treatment to improve social abilities in children with autism. So we set out, and this was in collaboration with one of my departmental colleagues and a child psychiatrist, Antonio Hardin, here at Stanford, to launch a double-blind, randomized, placebo-controlled treatment trial. And so we had two main aims. The first one, because nobody had ever studied vasopressin in the context of autism before, we wanted to make sure that vasopressin was safe and well tolerated across this 4-week trial. And so we assessed dropout rates from the trial. We monitored vital signs. And we assessed clinical chemistry, blood labs. We monitored heart function through electrocardiogram, and then also evaluated side effects and adverse events. And then we also wanted to ask whether this 4-week treatment improves social abilities in children with autism. And so we had multiple measures. One was the social responsiveness scale, which was a parent report measure. And then we had additional outcome measures, including clinician evaluations. And then we had children themselves perform objective laboratory-based social cognition tests. And what we found was that after 4 weeks of blinded vasopressin treatment, it was safe and well tolerated. So specifically, no vasopressin-treated patient dropped out of the study. There were no differences in adverse events or side effects between drug and placebo-treated individuals. And we saw no significant changes in vasopressin-treated patients' vital signs in any of their blood labs or on heart function as measured by electrocardiogram. And then, encouragingly, we saw that 4 weeks of vasopressin treatment improved core social symptoms, and we were able to see this improvement based on parent ratings of their observations in children's improvement in social abilities over this 4-week period, clinician evaluation, as well as child performance on 2 tests. So one was the Reading the Mind and the Eyes test, which asks individuals across multiple trials to ascertain what somebody is feeling. And so here's an example here. What emotion is this individual feeling? And so vasopressin was able to improve performance on the Reading the Mind and the Eye test, as well as recognizing specific emotions on still faces. So then I want to return to the question that I posed at the beginning of my talk, the title of my talk, which is vasopressin the key to unlocking our understanding of autism? And so we particularly were interested in these 2 social molecules that are closely related but distinct in their functions. And so what we found across these studies was that when we looked at both blood and cerebral spinal fluid, the thing that was across multiple studies in rhesus monkeys and children diagnosed with autism and infants who then subsequently developed autism many months to years later, that the thing that linked across all these different studies was low cerebral spinal fluid levels of vasopressin. And what I didn't have time to talk to you about was that there has been work in the oxytocin field, and there was a large study that was conducted several years ago now and published in the New England Journal of Medicine that showed that while intranasal oxytocin treatment was safe, it did not improve any behavioral features of autism. So suggesting that oxytocin may not be involved, at least in autism the way that we are conceptualizing it here. And in summary, again, we've published this initial pilot trial showing that vasopressin given intranasally did improve these core social functioning difficulties in children with autism. And currently, we've just completed one of the largest single-site medication trials for autism to date, where we gave vasopressin for 8 weeks to a cohort of greater than 100 children. And we will be reading these results out sometime this year. And so just to summarize, It's very clear that vasopressin has been implicated as a key factor in the biological basis of autism. And it may be that we can give intranasal vasopressin to improve social functioning in individuals with autism, allowing them to connect and engage in social interactions more effectively. And so then I want to just conclude by thanking my collaborators, various mentees, research assistants, and technicians, the children and their families for participating in our research. There are various funding entities, and you for your attention.